Hello everyone. In today's video, we will be learning about chapter 5, molecular basis of inheritance. First, let us see what is molecular basis of inheritance. Okay. So first, molecular basis of inheritance is the study of gene heredity, and genetic variation which will explain how our offspring will look similar to the maternal or paternal features. That is what is called as molecular basis of inheritance. It is a study of genes, heredity and genetic variation in order to look into the similarity between the maternal or paternal features and the child's similarity of genetic variation. Got it? So next is, what is nucleic acids basically? Nucleic acid. Nucleic acid is of two types. One is DNA, the other one is RNA. DNA means deoxyribonucleic acid. RNA means ribonucleic acid. Got it? So DNA will act as a genetic material in most of the organisms. Questions may come. Which acts as a genetic material in most of the living organisms? DNA is the genetic material in most of the living organisms. Whereas RNA is also a genetic material, but it has got many functions too. Like what? It acts as a genetic material, it acts as an adapter, it acts as a structural, it acts like a catalytic molecule. These much features it has got. Got it? So, in this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA from DNA, transcription, genetic code that determines the sequence of amino acid proteins, the process of protein synthesis, translation, and elementary basics of their regulation. This is what is all that we are going to see in this chapter. Got it? So the determination of the complete nucleotide sequence of the human genome during the last decade has set into a new era of genomics. So about which we are going to study. We are going to study about the new set of genomics that has been arrived in the last decade. This is in the past 10 years. Now let's learn about the DNA. Genetic material controls the formation and expression of traits in an organism. We know that, right? It can replicate and pass on from one generation to another. DNA, which is deoxyribonucleic acid, will represent the genetic material of an organism which contains gene and is responsible for passing down of the characters from generation to generation. So, how does the DNA occur? Let me give you the mind map. Occurrence of DNA. How does the DNA occur? First is prokaryotes. In prokaryotes, how does the DNA occur? And in eukaryotes, how does the DNA occur? First, let's see in prokaryotes. In prokaryotes, DNA is circular, naked, and found in cytoplasm. Whereas in eukaryotes, DNA is linear. DNA is linear and is found in nucleus. In subjective point of view, this is a very important question. Like in your school exams. The occurrence of DNA, prokaryotes, eukaryotes. In prokaryotes, how does the DNA occur? DNA is circular in shape, it is naked and it is found in cytoplasm. So let me draw the diagram so that you can understand it much more clearer. DNA is circular. In prokaryotes, DNA is circular. Chromosome, plasmid, they are naked and found in cytoplasm. This is what in prokaryotes. In eukaryotes, In eukaryotes, it would look like this. If this is a cell, this is a cell, this is the nucleus, this is the nucleus. If I zoom into the nucleus and count the chromosome, like in here, if I zoom in the chromosome and look into the particle over here, I'll find the DNA here. Like this. And this is the DNA. Got it? This is how the occurrence of DNA occurs. So now, how do we find the length of the DNA? Length of the DNA is usually defined as a number of nucleotides. It is the number of nucleotides. So a pair of nucleotides is known as base pair. Then I ask you, what is base pair? What is base pair? It is the pair of nucleotides. Pair of nucleotides. When two nucleotides combine, it is known as base pair. And how much number of nucleotides is present is the length of the DNA. It is the length of the DNA. Got it? So how do you find the length of the DNA? It is the number of nucleotides present. What is base pair? It is a pair of nucleotides. Got it? Coming to the next point, we have length of DNA in different living organisms. Like you can take the organism, I will just give you a tabular column, organism, and here we have the length of the DNA. So organism, bacteriophage, 5 into 174. What is the length of it? Length is 5,386 nucleotides. 5,386 nucleotides. So organism 2, it is bacteriophage lambda. Bacteriophage lambda, how much does it have? Bacteriophage lambda has got 48,502 base pairs. Okay, 48,502 base pairs. Base pairs. Okay. So now coming to the next is the Escherichia coli. E. coli. How much has E. coli got? It is 4.6 into 10 to the power of 6 base pairs. Next is the haploid content of human DNA. How much has the human got? We ourselves has 3.3 10 to the power of 9 base pairs. Got it? But it is for what? It is for human haploid content. If it is for diploid, it is 6.6 into 10 to the power of 9. Got it? Now let us look into the structure of polynucleotide chain. DNA is a long polymer of deoxyribonucleotides. This is what is DNA. Each and every ball here is deoxyribonucleotide. Okay, this one is called as DNA. The nucleotides are the building blocks of nucleic acid. They may ask you in MCQ. Nucleotides are building blocks of nucleic acid. Got it? So next is nucleotide has got 3 components basically. Nucleotide has got 3 components. First component is nitrogenous base. The second component is pentose sugar. And the third component is phosphate group. How will it look like? This is the phosphate group, right? Phosphate group along with the sugar. So the sugar. Here I'm taking, in case of DNA it is deoxyribose, in case of RNA it is just ribose. So as I'm taking DNA, then let me consider deoxyribose sugar. This is what this is called phosphate group, phosphate molecule. Phosphate molecule, deoxyribose plus this is what is called as nitrogenous base. In this case, I am considering adenine base. So this will all together it will form like this. Phosphate molecule attached with the sugar, attached with the adenine group, and this overall thing is called as nucleotide. In this case, it is adenine nucleotide. Got it? This is what is all about the structure of polynucleotide. Okay? Structure of polynucleotide. Coming to the next point, we have got nitrogenous base. Nitrogenous base are simply a nitrogen containing heterocyclic molecule that has the same chemical properties. There are two different types of nitrogenous base. So, nitrogenous base is a nitrogen containing heterocyclic molecule. Nitrogen containing heterocyclic molecule. It is of two types, purine and pyrimidine. First, let's differentiate what is purine and what is pyrimidine. Purine is basically larger size nitrogen containing biomolecule. Larger in size. Larger size nitrogen containing. Pyrimidine is smaller size. It is the first differentiation. Second differentiation is purine is a 9-membered structure. Is a 9-membered structure. Okay? Whereas pyrimidine is a 6-membered structure. 6-membered structure. Next. Purine is a double-ringed structure. Double ring. Whereas pyrimidine is single ring. Single ring. Okay? Purine contains 4 nitrogen atoms. 4 nitrogen atoms. Like in the positions 1, 3, 7, 6. Whereas pyrimidine contains only 2 nitrogen atoms in 1 and 3. Purine is of 2 types. Adenine, guanine. Pyrimidine is of 3 types. Thymine, cytosine, uracil. You can remember this by a mnemonic. For purine, you can remember pure agmark. Purine, adenine, guanine. Okay. Now, for some more clarity, let me give the diagrams of types of purines and pyrimidine. It is not that much important, but just see, watch it. Okay. So, adenine. How does adenine molecule look like? This is what it will look like. So, here, benzene ring, nitrogen, NH, NH2, N. It is how adenine will look like. Now coming to purine, how will this is all purine, right? Adenine is a type of purine. Let's see guanine also. Guanine is also a purine. So we saw first type of purine, adenine now. Now let's see how guanine will look like. Again, similar to adenine, but here no benzene ring. Instead, we have the same NH component. double bond N, NH2, here also NH2, here N, nice. This is what is called guanine. Now we saw how adenine will look like, how guanine will look like. Now let us look at pyrimidine. Right? How will pyrimidine look like? This is how thymine will look like. Thymine, NH2, N, NH, double bond O, thymine. This is how cytosine will look like. Cytosine, N3H, double bond O, NH, double bond O, NH. It is cytosine. Next we have uracil. Uracil. This is how uracil will look like. Now have you got an idea how purines will look like and how pyrimidines will look like? First is adenine. Second we saw guanine. Third we saw pyrimidines. In that we saw thymine, cytosine and uracil. Got it? So now let us get deep into the concept of pentose sugar. Let's see the individual components of nucleotide. First component, what did we see? It is pentose sugar. So let's dive deep into it. Nucleic acid contains a 5-carbon sugar molecule that binds nitrogenous base. Nucleic acid contains 5-carbon sugar molecule that binds to nitrogenous base. Right? In case of RNA, the sugar is ribose. In case of RNA, the sugar is ribose. In case of RNA, the sugar is ribose. However, in case of DNA, the sugar is ribose sugar has H atom instead of OH with 2 carbon position. Hence, it is called deoxyribose. I mentioned this earlier itself, right? Before 2 slides itself, I explained. Here, one oxygen will be missing. That is why it's called deoxyribose. Like a normal ribose in RNA will look like this: O, H, OH, H, H, OH, H, OH, OH. Right? In all the edges we have OH in RNA ribose sugar. But if we consider deoxyribose, OH, 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 OH will be there. But in case of deoxyribose, what will happen? One oxygen will escape and only one hydrogen is present. So one oxygen is being removed. That is why we call it as deoxyribose. Deoxyribose. Got it? One oxygen is missing. Now let us see bonds in DNA. There are 3 types of bonds found in DNA. First bond is hydrogen bond. The second type of bond is glycosidic bond. Third type of bond is phosphodiester bond. Phosphodiester bond. Now let us look deep into these 3 types of bonds. The first type of bond, hydrogen bond. Hydrogen bond in a DNA molecule, purine will bond with pyrimidine through hydrogen bond. Purine will bond with pyrimidine, and this bond is called as hydrogen bond. So basically, in between adenine and thymine, there is a double bond, whereas in between guanine and cytosine, there is a triple bond. Got it? This is what is all you need to know about the hydrogen bonding. So next we have glycosidic bond, a bond between a nitrogenous base and a pentose sugar. Bond between nitrogenous base and pentose sugar. How can you remember? Glyco. Glyco means what? Sugar. So with sugar you are bonding a nitrogenous base, that is known as glycosidic bond. So the one carbon of the sugar molecule is bonded with nitrogen atom. Therefore we can say it as it is bonded with nitrogen atom. Therefore we can call it to be N-glycosidic bond. N-glycosidic bond or glycosidic linkage. Got it? Coming to the glycosidic bonds are of glycosidic bonds of nucleoside and of DNA and RNA. First is adenosine, deoxyadenosine, next we have guanosine, deoxyguanosine, next we have cytidine, deoxycytidine, next we have deoxythymidine, these are different types of N-glycosidic bonds. Now we saw different types of N-glycosidic bonds. Coming to the last, which is phosphodiester bond. Phosphodiester bond. Adjacent nucleoside pairs are connected together to form a long polynucleotide chain via phosphate group. These groups are attached to hydroxyl groups at C. Nucleoside pair is there, no? Adjacent, which is nearby nucleoside pairs are connected together to form a long adjacent nucleotide pairs are connected together to form a long polynucleotide chain via phosphate groups. Okay? So these groups are attached to hydroxyl groups. Hydroxyl groups at 5 carbon sugar molecules and at 3 carbon sugar molecule of the next sugar molecule. 5 carbon and 3 carbon of the next sugar molecule. The bond between the phosphate group and the hydroxyl group is termed as ester bond. So, bond between phosphate group and hydroxyl group is known as ester bond. It's called as ester bond. Hence, these bonds are called as 3,5 phosphodiester bond. Got it? Now, theoretically, I explain some content now. I will show this in case of diagram how it will look like. So, let's see that. Okay? First, we have like this. Connected. Let's take adenine. Adenine is connected to thymine. And again a sugar, deoxyribose. Okay? Here we have got a bond. Got it? And again this bond is connected to the other sugar molecule. Again a bond. It will continue so on. Like this. And now we have guanine here. Guanine will form a triple bond with cytosine. Right? Again a deoxyribose sugar. Again a bond. Again we have got thymine here. Adenine, guanine, thymine, cytosine. So thymine will form double bond with adenine connected to a deoxyribose sugar and bond. Cytosine will form triple bond with glycogen and again a bond. This is how the molecule hydrogen bond will form. Here I have used a dotted line, right? Now highlighted in red color. It is what is called as hydrogen bonding. Got it? Red color is the hydrogen bonding. You saw diagrammatically now how the hydrogen bonding will be formed. Clear? Now we are clear with the hydrogen bonding. Now let us get into the topic of glycosidic bond. How glycosidic bond is formed? Already I told you, it is a bond between nitrogenous base and the pentose sugar. So nitrogenous base is there. Nitrogenous base. Nitrogenous base. And connecting this with the pentose sugar. This connection between the nitrogenous base. This is what this is. Nitrogenous base. And this is what down here it is pentose sugar. Then what is this? This is a bond connection, right? The connection. What is this connection called? This is bond. What bond? N-glycosidic bond. Now let us see how a phosphodiester bond will look like. So first let us take a molecule like this. Okay. Already I have told phosphodiester bond is the bond between the adjacent nucleoside pairs. Adjacent nucleoside pairs connected together to form a long polynucleotide chain via phosphate group. These groups attach to hydroxyl group at 5-carbon chain. 5 carbon of one sugar molecule and 3 carbon of the next sugar molecule. 5 carbon of one sugar molecule. So now let's take this is the 1, 2, 3, 4, 5. And again I am taking another sugar molecule. 1, 2, 3, 4, 5. Got it? I have taken 5 dash here and 3 dash here. And I am going to connect the 3 carbon sugar molecule and the 5 carbon sugar molecule. Now I have connected the 3 carbon and the 5 carbon. Here you could spot a ester bond. This is a ester bond. This is also ester bond. These two ester bonds together is known as phosphodiester bond. Phosphodiester bond. Therefore, we can also say that to be 3,5 phosphodiester bond. Now let us see DNA double helix model. In 1869, Friedrich Michener discovered that DNA as an acidic substance present in the nucleus and it is called nuclein. Rosalind Franklin and Morris Wilkins gave the X-ray diffraction pattern of DNA. In the year 1869, Friedrich Michener he discovered DNA as an acidic substance. He told that DNA is an acidic substance. He found that. Friedrich Michener. Okay. In 1869. And acidic substance present in nucleus. And he also told that DNA is an acidic substance which is present in nucleus. And he called it to be nuclein. Nuclein. Got it? Rosalind Franklin and Morris Wilkins gave X-ray diffraction pattern of DNA. Rosalind Franklin and Morris Wilkins. Morris Wilkins gave the X-ray diffraction pattern of DNA. X-ray diffraction pattern of DNA. Okay. So in 1953 only, Watson and Crick proposed a double helical model of DNA. Until that, they didn't know how the DNA will look like. Only in the year 1953. Okay. Two people. Their names were Watson and Crick. These two people gave double helical structure of DNA. Double helical structure. When they presented this model, it was too big. Actually, DNA is a very molecular structure. Right? Very small structure. But how did they even give that much big model? Only because of the X-ray diffraction which was given by Rosalind Franklin and Morris Wilkins. Okay. So this was based on the findings of Erwin Chargaff, about which we'll learn later. This is called the Chargaff's rule. So what did Erwin Chargaff pointed out in the particular DNA molecule is that total number of purine is always equal to the total number of pyrimidine. Total number of purine is equal to the total number of pyrimidine, which is adenine plus guanine equals cytosine plus thymine. Okay. The number of adenine-thymine bond differs from the number of the guanine and cytosine bond, but the base ratio is constant. Now, number of bond is different, but the base ratio A plus T by G plus P will always be constant. Got it? It is always constant. Let me give you a note point. Phosphodiester bond is in the backbone and hydrogen bond between the bases make the DNA a double helical structure. Okay, the phosphodiester bond is the backbone and the hydrogen bond between the bases will make the DNA a double helical structure. Got it? So how will this model look like? So basically, the DNA strands are anti-parallel in nature. Anti-parallel. Like this. If one structure is like this, starting from 5 primates to 3 primates, the other one will look like this, which is from 3 primates to 5 primates. Just opposite. Therefore, we can call it to be anti-parallel. Okay? Just like this. If linearly we are going to consider, it will look looking like this. Okay? This is the sugar phosphate backbone. As I told you priorly, here itself I have mentioned. Phosphodiester bond is the backbone. Okay? It is a sugar phosphate backbone connected like this. It will be connected like this. It is what? Cytosine, guanine. If cytosine is there, it will bind with guanine as I told. Adenine is there with thymine. Cytosine, guanine. Adenine, thymine. Like this it will get connected. So, anti-parallelly it will get arranged. This is DNA ladder. Later it starts coiling and will form like this. This is how the DNA structure looks like. Got it? Simple. This is what is called as double helix. This was the structure, the model which was proposed by Watson and Crick. Watson and Crick along with Wilkins received the Nobel Prize. Watson and Crick along with Wilkins. Who was Wilkins? Wilkins was the one who told that, who gave the X-ray diffraction pattern, right? X-ray diffraction pattern. This is what the helical model of DNA, they got the Nobel Prize. Okay, just an extra information. So let us see Watson and Crick gave. What are the features that Watson and Crick gave? First feature: two polynucleotide chains bound by a hydrogen bond. Two polynucleotide chains bound by hydrogen bond. On the hydrogen bond. Next, glycosidic and phosphodiester bonds. Glycosidic and phosphodiester bond. Next is DNA duplex around central axis. Next, they also gave about the polarity. As I told you now, it will be anti-parallel, they told. Okay, so polarity they gave, and complementary base pairing also they gave. Complementary base pairing, and also they gave anti-parallel strands. These were the conclusions that they gave. Watson and Crick gave. Now let us see the salient features of double helix. It is made up of two polynucleotide chains. Made up of two polynucleotide chains. I already told you it is made up of two polynucleotide chains where the backbone is constituted by. What is the backbone? Constituted by sugar polymer, sugar phosphate, and a bases inside. Two chains have anti-parallel polarity. Two chains have anti-parallel polarity. If one has a polarity like this, the other will have the polarity like this. Got it? So the bases in two strands are paired to hydrogen bond forming base pair. Bases in two strands are paired through hydrogen bond forming base pair. Okay, a purine always comes opposite to the pyrimidine. This generally approximates uniform distance between the two strands. They'll ask you a give reason question. Why does there is always a uniform distance in the helical structure of two strands of the helix? Because uniform distance is because of purine always comes opposite to pyrimidine. Therefore, there is a uniform distance. Got it? So next is the two chains are coiled in a right-handed fashion. Coiled in right-handed fashion. Right-handed fashion. The pitch of the helix is 3.4 nanometer and roughly 10 base per each turn. In each turn, like in each turn, there is 10 base pair. Like this, there will be 10. Okay. There will be 10 base pairs in each turn. Consequently, the distance between the base pair of the helix will approximately equal to 0.34. This is the distance. Got it? So if I have to give you a clear diagrammatical information, then it would look like this. how it will look like. So first, if you see, this is the whole distance, right, from this to this. One whole turn. This is one turn, this is one turn. So first, this is one turn. The complete turn. One whole this turn and one whole this turn. Together it is 3.4 nanometer. Right? Together it is 3.4 nanometer. Whereas between one hydrogen bond and the other hydrogen bond distance is 0.34 nanometer and the whole width is 2 nanometer. Got it? So last point is plane of one base pair stacks over the other base pair. One base pair will stack over the other base pair. So because of this only, in addition to hydrogen bond, it provides the extra stability. What is extra stability? Now let us look at types of DNA. Types of DNA. DNA can be categorized on the basis of handedness, like right-handedness and left-handedness. Okay, it can also be classified on the basis of number of strands, how much strands has. So DNA is mostly double-stranded. So some viruses may even have single-stranded DNA. What is shape also? On the basis of shape also you can classify DNA. Eukaryotes have linear DNA, as I already told you, and while prokaryotes and semi-autonomous organisms of eukaryotes and all will have the circular DNA. I told you, right? Before itself I told you, right? In the occurrence of DNA I told you. So now let us see the central dogma. Central dogma. It is a theory, okay? That is what we are going to see now. Francis Crick proposed. Who proposed central dogma? It is Francis Crick. He proposed central dogma on molecular biology. It represents a scheme how the information flows. How the information flows. This is how it will flow. DNA is there now. It will get replicated. Later it just converts into mRNA. Later it gets transferred into protein. This process is called as transcription. Transcription. This process is called as translation. Got it? So, process of copying DNA into mRNA is transcription. Process of synthesizing protein from mRNA sequence is called as translation. This is what is called as central dogma. Central dogma of molecular biology given by Francis Crick. So, let me give you one note point here. In some viruses, the flow of information is in reverse direction also. That we will call it as reverse transcription. That is, from DNA, from protein it will form RNA, and from that it will form DNA. It is called as reverse transcription. And reverse transcription examples are HIV and hepatitis B. HIV and hepatitis B. Reverse transcription. Let me write it here. Reverse transcription can also occur in some of the virus. Transcription can also occur in some of the viruses. Now let's get into the concept of packaging of DNA helix. So, taken the distance between the two consecutive base pairs. We know the distance between the two consecutive base pair equals 0.34 nanometer. Or I can also write this to be 0.34 into 10 to the power of minus 9 meter. Got it? If the length of the DNA double helix in a typical mammalian cell is calculated as, we want to find the length equals number of base pairs into distance. Like a 6.6 into 10 to the power of 9 into 0.34 into the power of minus 9, you will get it to be 2.2 meters. This is how you should find the length. Got it? Next, in order to accommodate a DNA molecule inside a cell, it needs to compact it and package. So, see, you yourself think, DNA, if it is 2.2 meter long, how can it get accommodated into such a small nucleus of 10 to the power of minus 6 meter? It has to be accommodated, right? It is Nowhere possible. So how it could be possible is that it has to be highly coiled. Highly coiled. Like this it has to become. Like very small it has to become. Right? So it has to be compacted and packaged. Compacted and packaged. Right? Compacted and packaged. In order to accommodate a DNA molecule inside a cell, that it needs to be compacted and packaged. Histones are positively charged proteins which are rich in basic amino acids like lysine and arginine. This helps in binding on negatively charged DNA backbone. There are 5 types of histone proteins: H1, H2A, H2B, H3, H4. Okay? So basically, histones are positively charged proteins. Let me write that. Histones, positively charged protein. And it has to be surrounded by DNA which is negatively charged. So now basically, DNA molecule coiled around the histone octamer forming the nucleus of core. What is histone octamer? Histones usually contain amino acid residue left over called arginine and lysine. They both are also positively charged, arginine and lysine. So 8 histone molecules come together. 8 histone molecules come together to form histone octamer. And then they are wound over by negatively charged DNA. Okay? This whole structure is called nucleosome. Got it? So, this can also be drawn like this beautifully. 1, 2, 3, 4. 1, 2, 3, 4. Wound by DNA. This is core DNA. Core DNA. And the histone proteins are inside. Okay? This is what is called as nucleosome. DNA molecules coiled around the histone octamer form the nucleosome core. Multiple nucleosome cores are joined via linker DNA. This DNA histone complex appears like bead on string. It will appear like a bead on string. Okay? Bead on string. Like this it will appear. If you view it under the electron microscope, it will appear like this. A bead string is coiled to form a cylindrical coil of solenoid having a 6 nucleosome per turn. 1, 2, 3, 4, 5, 6. 6 nucleosomes per turn. Okay? Which forms a solenoid of 30 millimeter of diameter. 30 millimeter. It looks like a solenoid, right? Okay? It will undergo further supercoiling to produce the chromatin fiber. Chromatin fiber. Packaging of this chromatin at higher level, it will require additional set of protein. Higher level, it requires additional set of protein. Set of protein. And what is this additional set of protein called as? Additional set of protein is called as non-histone complex protein, which is NHC protein. Non-histone complex protein, NHC protein. Non-histone chromosomal. Chromosomal also we can say non-histone complex or chromosomal. Chromosomal protein. Okay. Non-histone protein will play a role in organization and compaction of chromatin chromosomes. Higher order of the chromatin structural strength is of 2 types. I am writing this to be in 2 types. First type is euchromatin. The second type is heterochromatin. Heterochromatin. Euchromatin, heterochromatin. First in euchromatin, it is relatively loosely packed. Loosely packed. Heterochromatin is tightly packed. Euchromatin is darkly stained. Sorry, lightly stained. Euchromatin is relatively loosely packed area of DNA with RNA or protein molecule can access and copy the information stored in the form of protein. Whereas heterochromatin is a part of DNA that highly contains the chromatin structure. RNA or protein molecules cannot usually access the heterochromatin area, and therefore the information in such parts are not passed on to form the protein. Okay, did not form any protein. And euchromatin contains actively expressing genes, transcriptionally active, whereas heterochromatin is inactive. Euchromatin are lightly stained, whereas heterochromatin are darkly stained. Euchromatin will look like this, bead on a string, like this. Whereas heterochromatin will look like this. Okay? So now search for genetic material. So they wanted to know which is the genetic material. Until that, they did not know which was the factor which was carrying the genes to the next generation. Today, in today's generation, we know it is DNA. Everyone in the road will say, if you ask, go and ask someone, what is the genetic material responsible for the transfer of genetic information? Obviously, everyone will say DNA. Everyone would obviously know about DNA and all, and the genes and all. But in that generation, they were unaware of which was the genetic material. They didn't know. Because of these experiments only, we came to know that DNA is the material which was responsible for the transfer of genetic material. Okay. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search for the chromosomes located in the nucleus of most of the cells. There was a number of experiments that provided conclusive evidence that the chief genetic material is nearly all life forms are the nucleic acid, mainly the DNA. Right? What was that experiment which confirmed that the DNA was the genetic material? First experiment was Griffith transformation experiment. Griffith transformation experiment. Next is Hershey-Chase. Hershey-Chase. Okay? First, let us see the Griffith transforming principle. The principle which he gave is, in 1928, Frederick Griffith was a scientist. He performed the first experiment suggesting that bacteria are capable of transforming the genetic information through a process called transformation. Okay, in Griffith's experiment, he used two strains of pneumococcus. Pneumococcus, other name is Streptococcus pneumoniae. Or we can also call it it is Diplococcus pneumoniae. I told you already, right? Diplococcus pneumoniae. Okay, bacteria which infect the mice. Type 3 S smooth, which was virulent, and type 2 R rough strain, which was non-virulent of the host. While the 2R strain did not have the protective capsule, it was defeated by the host immune system. Non-virulent R strain does not cause pneumonia. Virulent S strain will cause pneumonia. Okay? Things we need to know before studying this experiment are, he used pneumococcus, Streptococcus pneumoniae, which is the bacteria responsible for causing pneumonia. Okay, this is a lung disease, right? So, for this, there are two types of bacteria, pneumonia bacteria. In Diplococcus pneumoniae itself, there are two types. One is of S strain, and the other is of R strain. S strain is usually shiny, smooth layer, which is virulent in nature. What is virulent? Virulent means fatal, which will lead to death, which will cause pneumonia. If R strain is rough strain, it does not cause pneumonia, is not virulent. We can also say it to be non-virulent. Okay, S strain is dangerous. Remember, it is dangerous. R strain is not that much dangerous. Okay. Now let's learn the methods of Griffith's experiment. Griffith carried out in the following setup only. The first set is the alive and virulent S strain bacteria was injected into the mice. The alive and virulent, non-virulent R strain bacteria was injected into the mice. The virulent S strain was killed by heat and then injected into the mice, and the virulent S strain was killed by the heat and then injected along with the alive virulent R strain into the mice. So observations are, Griffith observed that the live pathogen could kill the mice, but a dead, heated pathogen could not. However, when a dead pathogen was injected along with the live R strain, its virulent genes got transferred to the R strain, turning the avirulent strain virulent. Thus, the donor's genetic material could transform the recipient. Let me explain this with the help of this diagram. See, methods of Griffith experiment, observation of Griffith experiment. First, he took the rough strain, which is non-virulent, R strain, as I told you, and he injected into this mice. Okay, after he injected, the mouse lived, it was alive. So, he verified that R strain is not that dangerous. And coming to the smooth strain, virulent, when he injected, the mice died. Okay, when he injected, the heat killed the smooth strain. That is, he took the S strain and he heated, what will happen? The layer, outer layer gets damaged and it would form like this. When he injected this into this mice, it was still alive, which says that the heated S strain did not affect it, and it is non-virulent. And in the next case, he took the rough strain and the heated S strain, and he injected this into this mice, and the mice died, which said that this smooth strain has still got some genetic material inside, and which was transferred into this mice. Because of that only it become virulent and it died. Okay, this is all about the Griffith experiment. Now let us see the biochemical characterization. Biochemical characterization. However, even though Griffith found out that there was some genetic material just getting transferred, he could not find the biochemical characterization of the transforming principle. So after Griffith, there came three scientists. Their names were Avery, MacLeod, McCarthy. These three scientists purified proteins, DNA and RNA from heated S strain. They took heated S strain bacteria alone, and they purified three components: purified RNA, DNA and protein. Now they checked individually which one was causing that virulent factor. They took individual RNA, they took individual DNA, they took individual protein, and then injected to again check which of these three factors was actually transforming that hereditary material. Okay, so when they observed that whenever the S strain DNA was not inserted into the R, the mice lived, significantly largely transforming. They also discovered that the protein digesting enzyme, proteases, when they put in the culture medium, they are having S type. In each and every culture medium, S type is there. Okay. So now inside this they have used proteases. Inside this RNA. Inside this DNA. They use this. In the first case, when they use proteases, all the proteins were not there. In the presence of no protein, there was no transfer of virulent factor and the mice lived. In case of RNA, if they use RNAs, all the RNA was not there. So therefore, there was no transformation of the genetic material. Again, the mice lived. But when they use DNAs, all the DNAs were killed. Therefore, the mice lived. In the first case, the mice dead. Why? Because only the proteases was not there. DNA was there. If DNA is there, the genetic material will get transferred. The mice will get affected and will die. In the second case also RNAs, in the use of RNAs, RNA was not there, but DNA was there. If DNA is there, the disease will come and the mice will die. But in the third case, when they use DNAs, DNA was not there. If there is no DNA, there will be no disease and the mice will live. This is what is all about the biochemical characterization of transforming principle. With this, with the help of these three scientists, we found out that DNA was the genetic material which transfers the material. transfers genetic material. So, this is all about this part 1 video. In next video, in part 2 video, we will start with Hershey-Chase experiment. Thank you.